Alright, so in this video we're going to continue where we left off last time by talking about the transduction step 2, signal transduction in more detail. So like we just briefly saw last time, the transduction step usually involves multiple steps, multiple pathways that can amplify a signal and produce a larger cellular response. Right? There's more opportunities for coordination and regulation using this multi-step process. So going back to this picture from last time and specifically looking at the phosphorylation cascade because it happens in most cells. So if you remember, this is the reception phase where a signaling molecule binds with the binding site of our receptor and causes it to become active. And the receptor becomes active, it activates a messenger molecule inside of the cell. And we're going to really look at this part today. We're going to look at this part now. So here it is. And basically what happens is that first activated relay molecule, it's usually a protein or an enzyme. What it will do is activate another protein or enzyme. And then that one will activate another and another and another on down the line. What these proteins are what are called protein kinases. And a protein kinase is something that phosphorylates, which means sticks a phosphate group, that's what this represents, that P, onto another molecule. So once this first one becomes activated by the receptor, it finds the specific protein kinase that it is able to activate in the cell, and it phosphorylates it. It sticks a phosphate group on it. And once it has become phosphorylated, it is now active and is able to phosphorylate the next one in the chain that it is specifically able to phosphorylate on down the line. Over time, these activated kinases lose their phosphate group. They become dephosphorylated, and then they switch back off. This prevents a signal from continuing even after the signaling molecule is not bound to the receptor anymore, because over time, these phosphate groups will fall off and inactivate the protein kinases. So that phosphorylation cascade, that's what we call one phosphorylating the next, activating the next, all the way down the line, will go until the last protein that has been phosphorylated or activated is the one that will actually perform the cellular response. The next thing I want to talk about was called second messengers. So that signal that started by having a ligand bind to the receptor in the signal transduction pathway, we can refer to that ligand as the first messenger. It came directly from the cell that was sending the message. A second messenger then is like a small non-protein, like water-soluble molecules or ions that spread throughout a cell by diffusion that can carry the same message and activate those protein kinases as the first messenger. So there's a variety of different secondary messenger particles that we can talk about. You won't need to know specific examples, but you should be able to say whether something is functioning as a second messenger. It is the one right after the receptor that goes and activates all of the next initial protein kinases in the cell. So as an example, CAMP, or what's called cyclic adenosine monophosphate, is called that way because it's based on ATP. adenosine triphosphate, is an example of a signaling molecule. So CAMP is an example. It takes that message from the receptor and activates those initial protein kinases to start that phosphorylation cascade, right? And once it is degraded, it can turn back into adenosine monophosphate and it can be regenerated. So there are many signaling molecules or receptors that trigger the formation of this CAMP, the secondary messenger. So it is really important in, for example, like G protein coupled receptors, which is just a type of receptor that sends a message in one specific way. And you'll look more at those in your assignment for today. Um, and its job is to really activate the protein kinase A, which is kind of the first protein kinase in that phosphorylation cascade. So linking the receptor to the first protein kinase in the phosphorylation cascade, that's the job of a second messenger, like CAMP. So you're going to see this diagram in your assignment for today that's going to ask you to look at G protein receptors more specifically. Um, G protein receptor, that's just a term for one specific way in which a signal transduction cascade can work. It's useful to study because over half of the medications that are prescribed have their primary mechanism of action by either uh, increasing the activity of or decreasing the activity of G protein coupled pathways. Okay, so this is just kind of a little overview, but you'll look at one in more detail in your assignment. Just notice how CAMP, cyclic adenosine monophosphate, is acting as a second messenger, relaying the message from the first messenger, the signaling molecule, to the first protein kinase in the phosphorylation cascade. IP3 is another second messenger. Like I said, you don't need to memorize it. I just thought it would be interesting to mention because it is responsible for opening and closing ion channels, like calcium ion channels, which are important in maintaining the ion balance within the cell. So, for example, that's how this cell is able to take in or move out and calcium and maintain the balance of that ion within the cell. As an example, a signaling molecule could telling the cell that, hey, you need to take in more calcium might activate this IP3 second messenger that could go and open an ion channel that will allow the diffusion of calcium ions in from the outside to the inside. I don't expect you to know, and the test doesn't expect you to know about any of these steps in here and those words. If you go on and take biochemistry, you'll learn all about them, and they're really fascinating and they're awesome. But really, we just need an, I just am using this as another example of what a second messenger does. It links this first signaling molecule to the phosphorylation cascade or to the response, the calcium ions going in, just like that. All right. That's kind of the last thing that I want to talk about the signal transduction pathway. The last thing I just want to talk about kind of feedback, okay? Feedback and examples with the human body. So let's talk about this process with the human body for once. So here's the human endocrine system or organs in the human endocrine system, which do a lot of long distance signaling. So this general model works for them. And here's some the major glands. All of these hormones here under the hormone column you can think of as primary messengers. Those are what's sent out in the blood to attach to a specific cell or cell type to get it to do something. And these list kind of what they want, what these hormones want the cell to do. To lower calcium levels or raise calcium levels, all right, like calcitonin and PTH. So think of these as kind of using that secondary messenger we just talked about, IP3, that opens and closes calcium channels. So here are a lot of examples of those in your own body. Here's some additional ones, like for in the pancreas and the adrenal glands. So just getting an idea of how these signaling molecules and signal transduction pathways are really, really important in our own bodies. Um, so as an example, um, when you experience something stressful, that might cause your endocrine system to send out a message to elicit a specific response from a bunch of your cells, like to take in more glucose and increase respirations and things like that. And so, in the case of stress, your hypothalamus stimulates both your nervous system to send a message to the adrenal glands to secrete epinephrine and norepinephrine, which will go out as first messengers into your bloodstream to cause cells to do something in response to the stress. And your hypothalamus will also tell the pituitary to send out its own message to tell the cortex of the adrenal gland to make two different types of corticosteroids that will have a different response on cells. So the way in which these signals actually propagate through your body, like this picture says testament to, can be really, really complex. I just thought it would be a good idea to give you an example. Lastly, I just wanted to talk a little bit about the feedback loops that these can create. So, for example, if your body needs to respond to an increase in blood calcium level, this increase in blood calcium level might stimulate the thyroid gland to release a primary messenger, a signaling molecule called calcitonin, which is then received in the bones and in the kidneys, and its message is passed through that signal transduction pathway to get the bones to put more calcium, gain more calcium, and the kidneys to reduce the uptake of calcium in the urine to help to lower the blood calcium level. So here we have feedback, which is called negative feedback. We'll talk about that more later too, which ends up responding to a change in homeostasis in a way that it returns itself to a medium level. And the same thing can be said when the blood calcium level is low. In that case, low blood calcium stimulates the parathyroid hormone to release PTH, which is this primary signaling molecule that stimulates the digestive system to uptake calcium and the kidney to uptake more calcium in the urine and bones to release calcium to help get your blood calcium back to where it needs. So these signals can be switched on and off and changed to help maintain homeostasis. That's all I really wanted to talk about with this video is just some examples, um, some feedback here at the end, and then talking a little bit more about the transduction pathway and second messengers.